This screencast will address information about proteins, and this is essential understandings, or knowledge rather, 2.A.3 and 4.A.1. So proteins are extremely varied in their structure, which reflects their diverse roles they play in cells and organisms. So we have here an example of what these models of what these proteins look like at the molecular level. So alcohol dehydrogenase is an enzyme which will help to metabolize, break down alcohol. Epinephrine is an example of a protein that is a hormone. This is also known as adrenaline. Collagen is a structural protein found in your skin, as one example. And then insulin is another example down here of a hormone as well. So you see that the structures they look all pretty and colorful here, but they are indeed a little bit different from one another. And that is going to reflect their structural difference is going to reflect their functional difference. So enzymes work differently than structural proteins and hormones, etc. So proteins extremely varied, lots of structures, lots of different functions based on their individual molecular structure. So now it's time to look at that molecular structure of proteins. All proteins, even though they have different functions, have something that is very, very similar to them. All proteins have at their base the monomers of amino acids. So this on the left here is an example of an amino acid. So if we go back and look at the title, I should go back here actually. Proteins are polymers. In a previous screencast, we talked about polymers and how you make a polymer from individual monomers. So proteins are examples of polymers. We also sometimes refer to them as polypeptides, and you'll see why in a little bit. They're constructed from amino acid monomers via condensation reactions. A previous screencast addressed this whole idea of condensation reactions. So we're going to see that in action right here. The amino acids, their name gives it away. What is an amino acid? Well, an amino acid has an amino functional group. Previous screencast addressed that. And it's an acid, so here's our carboxyl group on the other end. Then there's this central carbon atom right here. So all amino acids have this nitrogen, carbon, carbon base to them. So again here, nitrogen, carbon, carbon. On one end is the amino end, right? And so this is sometimes referred to as the amino, right, terminus, and then this is the carboxy end or the carboxy terminus, right, because it's describing this functional group on the right there, the carboxyl group. This central carbon atom right here always has this hydrogen bonded to it at the bottom down here. But then up here, this is what's known as an R group or a variable group, right, which will make each individual amino acid different from one another. So again, all amino acids have this nitrogen, carbon, carbon backbone, and this central alpha carbon right here has the R group bonded to it. So a little bit more about those R groups in just a minute. First, let's put these amino acids together. So on the right here in A, we see that we've already sort of bonded two of them together in a previous reaction. And that bond is very specifically located between the carboxy end of one amino acid and the amino end of another amino acid. This bond has a specific name. It is referred to as a peptide bond. Why? Because it is. So this peptide bond right there is this carboxyl group and this amino group very specifically. Now we're going to show you here on the right further a condensation of another amino acid onto this, this smaller polymer right here. Incidentally, going backwards, this smaller polymer, since it's two amino acids here, right, is sometimes referred to as a dipeptide. Okay. So what are we going to make here? We're going to stick another amino acid onto it. So let's just take a second and look. Here's our amino group, and here's our carboxyl functional group. And oh, hello, we're going to do a condensation reaction here because this is the way we have to put things together. Here's a hydroxyl from this carboxyl group over here. Here's a hydrogen from this amino group, and we can condense them and make this water molecule over here. And in the process, then we form a peptide bond, which is right down here. So by condensing that amino acid onto that dipeptide, we've now created a tripeptide here. Right? 3 amino acids long at the bottom, 1, 2, 3 amino acids long. Regardless of how long this thing goes, right, we're always going to have at one end, right, this amino terminus, right, refer back to over here, here's the amino terminus over here, and then at the other end, right, we're going to go nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, and at this end we have the carboxy terminus right there. So we have 3 amino acids, we have 2 peptide bonds, we're generating here a polypeptide. So again, now proteins are polypeptides. All proteins have this in common with one another, is that all proteins get assembled this way. So then what makes a protein different? What makes a protein different, what makes collagen, say, different than epinephrine is the order, right, of the amino acids. The order of the amino acids, right, is going to be important here. Now, what do you mean order? Well, I mean that this amino acid here is different than this amino acid because of these R groups, right? So now we go back up to this R variable group here. So there are 20 common amino acids. Don't freak out right now looking at this slide here because you don't have to memorize any of this, right? Amino acids are characterized by their R groups, right? It says do not memorize this. You're not going to be expected to know this. What you are expected to realize, though, is a couple things. One, that there are nonpolar ones, there are polar ones, and there are electrically charged ones, meaning amino acids. But if you look, all these amino acids have this nitrogen, carbon, carbon backbone, right? Highlighted in white here is what makes the amino acids different from one another. Right? So you have glycine, alanine, valine, etc. These are all nonpolar because their R groups are nonpolar. Right? If you're unsure about what polarity is whether or not this is going to be charged, whether or not this is going to have partial charge, and you should see the screencast on water, the water molecule, to refer or to teach yourself a little bit more about that polarity. At any rate, 20 different amino acids here, 20 different ones because of their R groups. Each one of these R groups is different. So the order of these amino acids is going to determine how this molecule is now going to fold in 3 dimensions. So we have different levels of structure of polypeptides. We have a primary level, secondary level, tertiary level, and a quaternary level. So what you see here on the left is the primary structure is just simply the sequence of amino acids, right, bonded together via peptide bonds. So this looks like a string of pearls here, and this here's an amino and the amino terminus, and then here, look, here's the carboxy end right here. So this number 129 right here, this means that there are 129 of these amino acids all linked together, right, via peptide bonds. So this is the primary structure, simply the sequence of the amino acids. The secondary structure of the proteins is how this thing is going to begin to fold in 3 dimensions, and we're going to do a little activity in class that'll help bring this to life for you a little bit more. But the secondary structure is going to be determined by hydrogen bonding between amino acids. Right? If you're unfamiliar with hydrogen bonding, refer to the water molecule screencast for a little bit review on hydrogen bonding. So hydrogen bonding between the amino acids, and we either get an alpha helix or a beta pleated sheet as the secondary structure of these. The tertiary structure is where it's really at here in terms of functionality of proteins, right, where they're actually going to gain their functional level. So tertiary level structures here, right, are determined by various bonds between R groups of amino acids. So this is really what you need to concentrate on and be able to know and describe and sort of understand, is that the protein gets its function. Collagen is collagen, insulin is insulin, because of the way that that thing is going to fold in 3-dimensional space. And the way that it folds in 3-dimensional space is determined by which amino acids are placed where. So this ribbon right here is supposed to be already the primary and secondary structures, but you're zoomed out a little bit. And what we're going to see is that the amino acid that is right here has this functional group on it. Well, this functional group happens to be a polar functional group. And this amino acid on down the line over here happens to have this functional group, which is also a polar one. So what happens is you get this hydrogen bonding between these two amino acids here, and that causes this, this kink right here. It causes this fold, right, in the ribbon to get in 3 dimensions. Also along the line, you have another amino acid there and another amino acid here. This amino acid has this nonpolar R group or side chain. Here, this one has a nonpolar R group or side chain. And here we get hydrophobic interactions, right? Van der Waals interactions holding this, keeping this fold in place. So right here we've seen one type of bond that holds the tertiary structure together. Here's a second type of bond that holds tertiary structures together. A third type of bond that's going to hold tertiary structures together, right, is shown down here, where this amino acid and this amino acid here, right, here's the amino acid, here's the R group of this one, and here's the R group of this amino acid. And these are fully charged or ionic side chains or R groups. And so the third type of bond that holds the tertiary structure together is are ionic bonds. The fourth kind is shown up here with the disulfide bridges. Again, there's an amino acid, there's an amino acid, here's the side chain, here's the side chain. These side chains are both sulfur bearing, and you have this little disulfide bridge in there, and this is the fourth type of bond. So again, the tertiary structure, hydrogen bonds between R groups, Van der Waals interactions between R groups, ionic bonds between R groups, disulfide bridges between R groups, or bonds between R groups. Most proteins are functional at this level, right? So once you get this thing folded, functional at this level. Other proteins are functional at maybe a quaternary level structure. So the next level of structure is determined by the bonding of 2 or more tertiary structures together. So here's an old friend, collagen. And you see that it's sort of this braided, looks like a hair braid, where each one of these pieces of the braid would be a tertiary level structure. So to get this collagen, you take 3 tertiary level structure proteins, and they sort of get wound together like that. Another classic example would be a molecule of hemoglobin. And here this has 4 subunits, and you can kind of roughly see them. Here's one subunit, here's the other subunit in that color, here's a lighter pink subunit, and then there's a darker pink subunit there. So there are 4 subunits here. Each of those would be a tertiary level, right? And then they're all sort of bonded together, right? So you have alpha chains, beta chains, right, there, etc. So hemoglobin, another classic example of a tertiary level structure protein. So these proteins, their function is determined by their structure. And here is just a summary slide of all of this. So here's your primary structure. Secondary structure are alpha helices, beta pleated sheets. Tertiary structure held together by 4 bonds, ionics, disulfides, Van der Waals, and hydrogen bonding. So there's 4 to review there. Quaternary level structure then is more, 2 or more of these tertiaries together. This protein folding on YouTube clip is available as a separate URL on Blackboard. I would recommend going to take a look at that. There's also another little thing on the Sumanis website about protein denaturing. So I'm going to talk about that right now, but then that other animation is a little bit better maybe than what I'm about to do here. So we have this normal protein, which would be a functional protein, and you see it, to us it looks like just a blob, right? This ribbon model here. So this is a ribbon model of the protein. Looks kind of like a blob, right? But this is functional at this, with this specific structure, right? So this is a functional one. Over here, this denatured protein is non-functional because its structure does not look like the one on the left. So when these proteins become denatured, it means that they are losing their structural integrity. So denaturing, right, is losing their structural integrity, meaning that the ionic disulfide van der Waals and hydrogen bonds of the tertiary structure are breaking. You get this denatured protein. This is non-functional. Right? You could renature it, in which case this thing would fold back up, and those bonds, the hydrogen, the van der Waals, the disulfides, and the ionics, again, would then reform. Right? But oftentimes, once you denature, it's very difficult, it turns out, in most cells to sort of renature that enzyme. Again, clicking on the Semanis link down there or looking at the link on Blackboard may help you with that. Visualize that. Do I want to talk about this? No, not right now. So this last slide here is just a place for you to sort of summarize what you just heard there. What kind of elements do you see? Right, so this is, this is an organic, right? So obviously you're going to have carbon and hydrogen and oxygen, but what else do you have? What are the monomers of proteins? What are the polymers of proteins? What sort of bonds or linkages do we discuss over here? And then some properties, structures, functions, or just random other things that you would need to know here about proteins. Okay? Questions? Bring them to class and let's talk about it.